Today, we concentrate on the locomotory organelles in protozoa, and along with that, the different mechanisms of locomotion in kingdom Protista. As you can see, the locomotory organelles include the pseudopodia, the flagella, the cilia, and the contractile structures. Now, let's move on to the different type of pseudopodia. I had already given you this the other day. Now, what are the pseudopodia? They are the structures which are formed by the streaming flow of cytoplasm. Now, pseudopodia, these are of different types. As you can see, the lobopodia structure here, you can see the philopodia, the podium, which looks straight, you can say needle-like, whereas in the next one, which is it is the exopodia, it is long filamentous type, whereas in the reticulopodia it is forms a sort of network. So let's move on to the next slide, and here you can see in detail those structures. Now lobopodia are the pseudopodia which are present in the amoeba. So here you can see these structures, they are lobe like pseudopodia, and they have you can see they are broad and they have rounded ends as in amoeba, and they are composed of both ecto as well as endoplasm, and this lobopodium moves with the pressure flow mechanism. So let's move to the next slide. Here you can see the philopodia. Now the philopodia, this also I showed you the example is euglypha. Here you can see these are more or less filamentous structures, and then the pseudopodia, you can say usually they are tapering from base, and they are pointed at the tip. As you can see here, these are tapering at the tips. And example I've already given you, these type of philopodia are composed up of ectoplasm only. So let's move on to the next slide. Now here you can see the reticulopodia. Now these reticulopodia, example here given is the, you can see this is L phidium, this is the reticulopodia. These are, you can say, filamentous structures which form a sort of network. That's why we use the term reticulopodia. And the they are you can say filaments are branched and interconnected, and they display two-way flow of cytoplasm. So let's move on to the next one. Here you can see this is the exopodia. Now exopodia, as I gave you that day, this is the, you can say, looks like the sun animalcule found in such organisms which are the sun animalcules. Example is actinophris, and here you can see these have you can say needle like straight pseudopodia. So these are the pseudopodia actually, and these you can say they contain a central axis, as you can see, central axial rod, which is covered by granular and adhesive cytoplasm is there. And exopodia displayed here, there is also two-way flow of cytoplasm. So exopodia are characteristics, these are found in the heliozoans. So let's move on to the we can say, I'll just wait here and then again come back. Now let's concentrate on the amoeboid locomotion. As you can see here, this type of locomotion is also found in the sporozoans, in the phagocytes. Of course, sarcodines are there, and as there are different people who are giving different theories, several theories have been put forward, and the names of such theories are, for example, there's a surface tension theories which was given by Butterfly. Year is given here. Then there's a ruling movement theory given by Jennings. Similarly, we have the walking movement theory which was given by Dellinger, but most accepted from all these theories is the amoeboid. In the amoeboid locomotion is the change of viscosity theory, which is called as a sol or gel theory. So this is what we are going to concentrate on. The sol and gel theory was first given by the scientist name is Hyman in 1917, and it was later supported by Pentin in this year. Similarly, by another scientist, Mast, then Rinaldi and John in different years. Now, this enjoys a wide acceptance, and the basis of this theory is the pseudopodia are formed and withdrawn due to cyclic viscosity change. Again, I'll repeat the word. The pseudopodia are formed and withdrawn due to cyclic viscosity change in the, you can say, consistency of the cytoplasm from sol to gel and from gel to sol forms. Okay, means sol is solution form, gel is, you can say, little hard, or you can say gel type of structure. So as you can see, this is a diagram which you have to make for the amoeboid movement after solvent gel theory, which was given by Mast. So here you can see, on the posterior side, you can see here this structure which is formed is called as a uroid. This is a pseudopodium, which is the movement, advancing movement of the pseudopodium, or the animal is going to move in this direction. And here you can see on the posterior side, this dotted structure which you can see is the plasma gel. So we know that there is ectoplasm here, and then we have the endoplasm in the center. So the ectoplasm, as you can see, is formed of the plasma gel. So there is plasma gel, and then there is plasma sol also, whereas the inner one is called as the endoplasm. All right, and then outside is the plasma lemma. And here you can see towards the posterior side, the plasma sol is getting converted to the gel is getting converted to the sol form. Gel is being converted to the sol form, and then this sol form is moving forward, and as it is reaching towards the direction of movement, it moves to the side, and you can say giving a sort of busting type of movement, and it moves further, and this is the place where the pseudopodia will be formed. So this is the diagram which you have to make. Let's just see in the theory part also what they are saying. According to this, the cytoplasm of amoeba consists of an outer layer of jelly like substance, and you can say, which surrounds a fluidy mass. So there is jelly on the outside and fluidy mass in the center, and this is the former is called as the plasma gel, and the later is called as the plasma sol. So it means that the fluid like part is the sol, and the jelly like part is the gel. From gel you can use the word jelly. And the locomotion, you can say, in this way is explained by these four processes. Let's see. Outermost thin elastic cell membrane, or the plasma lemma, becomes attached to the substratum. So in the beginning, the outer layer है जो इसका elastic cell membrane का, और the plasma lemma, this attaches some substratum को attach कर देगा. Then there is local partial, अब फिर क्या होगा, there is a local partial liquefaction of plasma gel at the anterior. उसके anterior end पे there is a liquefaction of gel से बन जाएगा, you can say liquefaction of the gel towards the anterior and to the sol form. This से क्या होगा, this causes the central, जो बीच में plasma sol is there, which is under tension, to flow for, वो आगे को flow करेगा, and force the plasma gel against this weakened force, जो एक weakened force बन गया है, so it is going to force it, which produces a bulge, which is actually the beginning of the formation of pseudopodium. So as the plasma sol enters the, you can say, the newly formed pseudopodium, at once it changes to plasma gel. So as soon as it reaches, changes to plasma gel around the periphery, thus forming, you can say, the gelatinized tube within which the plasma gel continues to flow forward. You can say now next, what happens on the posterior side, inner surface of the, you can say, plasma gel undergoes solation. So on the posterior side, solation takes place. So the vacuum which is created, or because the fluid has already moved forward, so solation takes place. Now you can say there is a constant flow of plasma sol, or the solution is maintained from behind forward towards the direction of movement. So thus the outer tube of the elastic plasma gel contracts. So outer tube is contracting and moves from in front backward. As it reaches forward, it moves from front backwards की तरफ जाएगा, in the direction of the movement. So this is actually the process, and we see that the plasma gel thus exerts a squeezing. There is a squeezing motion from the, you can say, sides and rear of amoeba, forcing the plasma sol to move ahead. So this is the type of locomotion which is found in amoeba. Now there is some organisms, for example, in sarcodines like mestig amoeba, they have both pseudopodia and flagella is there. So we can say it is a connecting link between what? Between the pseudopodial organisms and the one with the flagellum, flagellates and the sarcodines. Okay, so and you can say that these threads pad-like structures which are present when these organisms are called as the animal flagellates. We also call them zooflagellates. So we reach the term flagellum. ठीक है, flagella. So next locomotion will be with the help of flagella, which is found in the zooflagellates. Example is Trichomonas. Then we have the Giardia, which has 4 pairs of these flagella present. So their number is different in different organisms.